So I'm going to start talking about Unit 4 from this slide. Remember, you have to do and prepare the previous content for Unit 4. So in the earlier slides of Unit 4, the main idea in the earlier slides is basically telling you that bacteria, microorganisms, bacteria, they need nutrients. Yeah. Just like you need nutrients, yeah. You need carbon, so do they. You need oxygen, so do they. I mean, they're no different. They're alive, like you and I. So it's not a surprise. They need nutrients, yeah. And then nutrients need to come into the cell, of course. So that's where simple diffusion comes in play, facilitated diffusion comes in play, and active transport, and the endocytosis and the exocytosis and all that. And they have to deal with the environment, hypotonic, hypertonic, isotonic. They're exactly like your cells. They need food, they deal with the tonicity of the environment, and they have to bring in the food into the cell and maybe expel out waste. So that was the first part of the unit, and you will read up on that. So what's the second half? The other things that they need. One of those things that they need besides nutrients is a temperature, a adequate or optimal temperature. Yeah, just like you and I. What is our optimal temperature?
98.
98.6. I'm in Italy. I have no idea what that is. 37 Celsius. You see? 98.6 Fahrenheit. That's only here. Everywhere else in the world, it's 37 degrees Celsius. And in science, it's 37 degrees Celsius. So 37. Good. So you need a specific temperature, yeah? So do they. But the interesting thing about us, it's 37 Celsius. Now, do we die at 36 and die at 38? No, thank God. So we can handle a range, of course. Not too low, but not too high. So do they. No different. So bacteria, however, are different than us in another way. There are a whole different bunch of kinds of bacteria. Some of them like it hot. Some of them like it cold. And so forth, and all the things in between. So the ones that like it cold are given to you in the blue line. You do not need to remember the ranges, but I will tell you the optimals. Around 5 degrees Celsius or so, they are called psychrotrophs. Obviously, all these words are in your soft chalk, and they're also in your lab manual. Psychrotrophs. Sorry, no, erase that. Psychrophile. Psychrophile. All right, psychrophile are the 5 degrees optimal temperature. Psychrophile.
Is it Celsius, you said, right?
Celsius, yeah. When I'm talking temperature, it's Celsius, yeah. So 5 degrees Celsius, psychrophile. I misspoke. It's psychrophile, not psychotroph. It's the next one that's a psychotroph. So the next one, say around 25 degrees or so, optimal, that's the psychotroph. But it's also called psychotolerant. Either one is fine. So psychrophile, you're 5 degrees or so. Psychotroph, 25 degrees or so. And again, another name for psychotroph is psychotolerant. And then you have the ones that like around 37 degrees. They like it around 37. And these are your mesophiles, M-E-S-O, mesophiles. So the bacteria that live in us, they're mesophiles. Why? Because you just told me. What was our optimal temperature? 37. So the bacteria that live in you are mesophiles. You are a walking incubator for them. They love you in multiple ways. Now, there are some bacteria that like it a little hotter than that. And we're talking about, say, 70 degrees. Those are your thermophiles. Thermophiles. And there are some that like it hotter. And these are your extreme or extremophiles. thermophiles, and that's about 125. Extreme thermophiles or extremothermophiles.
What was the temperature for
About,
thermophiles?
for which one?
Thermophiles.
70, around 70. So let's do a review. The psychrophile, about 5 degrees, optimal. The psychrotroph, say 25 degrees. The mesophile, around 37.
That's us.
Huh? Say?
That's like in us.
That's us, yeah.
Okay.
And the thermophile, 70. And the extreme thermophile, around 125. Yeah. So all Celsius, yeah. It's all Celsius. Everybody good? Pretty straightforward? Alright, good. Let's turn up the heat. So, now, the other thing that bacteria will need, at least some of them, is oxygen. Others may say, oxygen? No, that kills me. So some of them will want oxygen. Others will say, no, 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 don't bring me near it. It kills me. So we are going to do this experiment in the lab, actually. This experiment right here. We're going to do it in the lab. The name of this media is fluid thioglycolate media. Just call it FTM for now. FTM media. But you're about to set it up. I think we're going to set it up on Tuesday. Fluid thioglycolate media. What is the purpose of this media? Let's say you have a bacterial species or a culture in your hand and you don't know, does it want oxygen? Does it use oxygen? Does oxygen kill it? You don't know anything about it, but you want to find out. What you're going to do is you're going to use this media, fluid thioglycolate media, because it tests for that. The FTM media tests for something called aerotolerance. Can it tolerate the oxygen or not? Aero in this case means oxygen. Aerotolerance. Does your bacteria tolerate oxygen or not? So how will you figure it out? You use the FTM media. But how do you do it? You inoculate the media. It's as simple as that. You take a sample of your bacteria that you're testing, and you simply inoculate the media. And you look a day or two later where the culture will grow, the location in this media. Now, do not confuse this with pellicle. Do not confuse this with sediment.
That's what I thought as soon as.
I know you were thinking that way. Call it experience. All right? Don't confuse it with that. The pellicle is because of mycolic acid. The sediment is because it does not have a flagella. Remember all that? And the
Yeah.
turbid. But we just used regular media when we did that. We just used media. So the pellicle, the sediment, the flocculent, the turbid, that's just using regular media. That's not this. This has a different purpose. This media is designed to test for whether your bacteria is consuming oxygen or not, or if oxygen is killing it. So it has to do with aerotolerance. So this is reducing media. This is media that does not have oxygen, at least initially. When I give this media to you, when I give this tube to you on Tuesday, it's not going to have oxygen in it. At the time of you inoculating it, it will not have oxygen, and there's a way to do that, and I'll talk about it. It will not have oxygen. But then you inoculate it, and the oxygen will come back. As you're incubating it, it will come back. Now the question is, how far will the oxygen go in here? Watch this. The oxygen is right here in the area where there's no media. There's a little gap behind this cap here. The air actually, it's designed for that. Air actually can seep in and enter the tube. That's why we tell you, don't hold the tube from the cap, because it's deceiving. You think the cap is hanging onto the glass. Barely. It's barely hanging onto it. It's not screwed on there. It just sits on there. And there's a little gap to make sure the air gets in. So some of these caps over time, they get looser. Then you go hold it like this, it just falls. And we've had a couple of those accidents, right? So you definitely have to watch out for that. But the idea is that this area here has air. So that's where the oxygen is. The thing about oxygen is it can diffuse through the media. It will. It'll go through the media, but only so far. It's not like carbon dioxide. Carbon dioxide can go through the media. Carbon dioxide can diffuse through water more readily than oxygen can. So oxygen can only go so far, but CO2 can go farther, you see? So if you think about it now, that means that the oxygen can only go so far, let's say here, as far as here. This area here will have oxygen, you see? That means this area here will not have oxygen. So this is the aerobic region, and this is the anaerobic region. So if you're a bacteria that is aerobic, that means you need oxygen. Where would you grow? On top. It looks a lot harder than it actually is. The hardest part is that to make sure you don't think it's pellicle and sediment. That's number one. This has nothing to do with that. It has to do with whether the bacteria wants oxygen or not. So you're going to use this special media. It's called reducing media. Removes the oxygen. You inoculate it. Then the oxygen comes back. Wherever the oxygen is, that's where the aerobic bacteria will grow. Because the aerobic needs the oxygen. That's why it's called aerobic. All right? Wherever the oxygen isn't, that's where the anaerobic will grow. Because the anaerobic cannot handle oxygen. Oxygen kills it. So it's going to grow on the bottom. Pretty straightforward. And then you got this weird one. Facultative anaerobe. My buddy. Why? Because as a microbiology student a long time ago, many decades ago, this thing drove me nuts. Nobody told me that a facultative anaerobe is actually an aerobe. What? Yeah, it's weird. Listen to this one. A facultative anaerobe is actually an aerobe. It means it actually wants oxygen, prefers oxygen. Facultative means if you give me oxygen, I'll use it. I actually prefer it. But if you don't give me oxygen, I'll still survive. See, this one cannot say that. The obligate aerobe means I definitely need oxygen. That's what the word obligate means. The facultative anaerobe says, if you give me oxygen, I'd prefer that. I'll grow better, but I will not die in the lack of oxygen. I am a facultative anaerobe. Do you remember, not too long ago, I told you every time you hear the word differential, replace it with what? Distinguish. And hopefully that helped you, right?
Yes.
Here's another one. How many times have you heard the word facultative in your life? 0? Never, right? How many times have you heard the word flexible? A lot. I hope. Otherwise my point doesn't make any sense. So I want you to replace the word facultative with flexible. Now it will make sense. I am flexible. You give me oxygen, I'll use it. If not, it's okay. That's the idea. Because that word facultative can be attached to a lot of things. It just happened that they attach it to the word anaerobe for today. So facultative anaerobe is a flexible aerobe. It's actually an aerobe. And that's what drove me nuts as a micro student a long time ago.
It prefers air?
Huh?
That's because it prefers
That's
air.
beautiful. That's why.
Okay.
Why is it an aerobe? Because it prefers oxygen. And how do you know? Well, look at it. It's growing better in the area where there's oxygen. Am I right? Compared to the area where there isn't oxygen. You know how long it took me to figure that out eventually as a student? A long time ago. because of this, in my eyes, why did they call it a facultative aerobe and get it over with? That's what they call it, so be careful with that. A facultative anaerobe is actually an aerobe, and the main reason why is it actually prefers oxygen. But really why is because in the presence of oxygen, we're more efficient in making ATP. So this guy makes more ATP in the presence of oxygen, that's why it can multiply quicker and more. Yeah, go ahead.
So what about the ones that do not prefer oxygen?
Oh, the one, they use other mechanisms to survive. Yeah, we'll talk about them. They're called anaerobes. We'll talk about them when we talk about cellular respiration. And you're gonna regret asking me that question. Go ahead.
In the facultative part?
Oh, we're talking about that one too. We're talking about all of them. We're gonna layer cellular respiration on top of all of those things. And we will have fun with them. Now you got the microaerophiles. Listen to this one. I am aerobic. I do use oxygen, but not atmospheric oxygen. You know the oxygen that you're breathing right now? That's called atmospheric oxygen, oxygen in the atmosphere. The percentage of the oxygen that you're breathing is around 21%. That's too high for these guys. It kills them. It's too much. So it says, I do want oxygen. I do use it. I do need it, but not at 21%. I need it in smaller amounts. You see? That's why they're called microaerophiles. They are aerobic, but in smaller amounts.
So
Yeah.
do they tend to like survive on below the surface of the earth?
They could, yeah. They're gonna survive in areas where there is less oxygen, of course, and they're not easy to grow, obviously, in the lab because you gotta control the oxygen concentration in special chambers and things like that. They're not easy to grow. The aerotolerant anaerobe. Listen to this one. This guy says, "Give me oxygen, I don't care. I don't use it. Give me oxygen, I don't care because it doesn't kill me either." So oxygen, it does not use oxygen, and oxygen does not hurt it. For the aerotolerant anaerobe, the oxygen is just there. It doesn't use it. It doesn't hurt it. It's just there. Now look at the subtle difference between an aerotolerant and a facultative anaerobe when it comes to the growth. Where is the main difference?
The top.
The top part. The
The
aerotolerant grows evenly all the way through. See, I don't care if I have oxygen, and I don't care if I don't. Whereas the facultative says, I'll grow in both, but I would rather have oxygen. Sounds good? So, technically speaking, I talked about the differences between the obligate aerobe and so forth. So the next slide I'm not going to go over because it's just another reiteration of the differences between obligate aerobe, facultative anaerobe, and so forth. But there is one more thing I need to talk about on this slide that's very important. Because you do this too. Are you breathing oxygen?
Yes.
I hope so. Hope for a long time to come. And if you are, I got news for you. You are producing this bad guy. This guy is called superoxide ion. You know, growing up when we were kids, we think Superman, he's coming to help me. You know, not this guy. He's coming to kill you. That's what this guy is doing. This is not a superhero. It's called supervillain, yeah. Superoxide ion. This is a charged oxygen. That's like a bullet going through your cytoplasm, damaging and destroying your DNA in your nucleus, the proteins in your cytoplasm, and if it passes by a mitochondria, it kills it, knocks it out.
Jeez.
You don't want this guy hanging around. But I got some bad news for you. You can't do anything about it. You're gonna have it. Your cell will produce superoxide ion. The question now is why? It is a byproduct of mitochondrial function. When your mitochondria is working to make what? ATP. A consequence of making ATP is making superoxide ions. You know what? We always say the mitochondria is like a power plant, am I right? Or a factory, right? Well, if you know, every power plant or factory produces waste. Every one of them. And sometimes the waste is toxic waste. This is the one that produces toxic waste. The mitochondria's waste is this guy. So there's only two ways of dealing with waste produced by a factory, for example. You go to the factory manager and you say, shut down. Close your factory, you're producing too much waste. Okay. That would not be good, because then the factory cannot make any money. And whatever it is that you're relying on the factory producing, you're not going to have. You don't want to close down the mitochondrial factory. Because what's the mitochondria making? ATP. You close it down, you shut it down, you kill that cell because it's going to lack ATP. So that's not an option. So what's the other option? Deal with the waste. We need this factory running. I know it produces waste, yeah. Deal with the waste. And that's what this guy does. Superoxide dismutase is an enzyme. It's an enzyme in your cells that basically deals with the toxic waste. That's the only solution. Because if you shut down the mitochondria, that cell is dead. So, any organism that uses oxygen, you are one of them, will produce superoxide ion. Now the question is why? Well, we said it's because of mitochondrial function. But that was the easy answer. Don't stress out with what I'm about to say. Because I'm going to see some faces right now go, what is he talking about? You mean I needed to remember that from Gen Bio? Yeah, but I'll teach it to you. Don't worry about it. Do you remember the electron transport chain?
Yeah.
Does it bring back good memories, I hope? No? Well, we're going to talk about it again in another unit or so.
It keeps coming back.
I know, it just keeps coming back. Wait and see. Watch this. Let me just a quick walk through memory lane for a real quick. But you don't need to know it in detail right now. We'll wait another week or so. All right? Complex 1, complex 2, complex 3, complex 4. Is there a complex 5? No. Better not be. All right. So 4 complexes. They're in your inner membrane of the mitochondria. In bacteria, they're in their cell membrane. Sounds good? All right. If you remember, the electrons went to complex 1, and then they went to complex 3, and then they went to complex 4, and then who got them at the end?
The ATP cell.
I'm about to hyperventilate. Oxygen. There you go. Let's try it again. This is guy doing it, right? Let's try again. The electrons went to complex 1, then they went to complex 3, then they go to complex 4, then they go to oxygen. That's why you're breathing, right? And then oxygen becomes what? Water. Don't be afraid. I will not bite you. Don't worry about it. Water. Very good. But then, in some occasions, the electrons can actually go to complex 2 from a different carrier. So complex 2 gets the electrons, then the rest of it is the same. Complex 2 to 3, 3 to 4, 4 to oxygen, oxygen becomes water. Now, if you know what I'm talking about, good. If you have no clue what I'm talking about, don't worry about it. Just let me finish this part. The whole reason why I'm saying this, and I'm going to teach it in detail in about a week or so, is to tell you what else can happen. The normal route of the electrons is through the complexes finally ending up with oxygen, becoming
Water.
water. But sometimes the complex, let's say complex 1, it does this weird thing. It says, every day I have to give the electron to complex 3. Every day. It's so boring. I'm going to do something different today. What is that? I'm going to give it to oxygen straight. Why do I need to do this? Hey, oxygen, yeah, here, electrons. Oh, bad news. You just made a charged oxygen. It cannot go directly to oxygen, but it does from time to time. Most of the time, it goes through the complexes, everything is fine. But once in a while, the electrons go directly to oxygen. You just charged this oxygen. Charged it to do what? To knock out your mitochondria, destroy your DNA, damage your proteins, causes you to age.
Oh God.
Now you have chronic age diseases, and as you get older, you get sicker. It's a lot of it is associated with this bad guy. So we're gonna call this guy the real bad guy. How many reals?
2.
2. There's a reason behind it. This is the real bad guy. You don't want him around, but you can't avoid it. He is around. So you thank God you have this guy, superoxide dismutase. It's an enzyme that converts the superoxide ion to hydrogen peroxide, H2O2. Now I get some students at this point and say, wait a minute. I remember in Gen Bio, yeah, that was a bad guy too. Yeah, that's why I call this guy real bad guy. Watch this. The superoxide dismutase takes the real bad guy and converts him to a real bad guy. Because this one can hurt you too. Remember, that's why we put hydrogen peroxide on wounds and stuff, to kill bacteria, to kill cells. So this guy can hurt you too. So we have another enzyme, catalase. You should be familiar with that one from Biolab, Gen Bio.
I've heard it.
Then you got catalase that takes H2O2 and converts it to 2 good guys. Look at that. From real bad guy to real bad guy to 2 good guys. Water and oxygen. Now that's not how you get your water. You don't rely on this to get water. You get your water by drinking it. And you don't rely on this for oxygen. You get oxygen by breathing it. What I'm saying is that these 2 enzymes work together to get rid of this real bad guy, and it turns it into 2 good guys. Nobody says no to water and oxygen. Is that amazing or what? So we have them. They're literally saving you. But as, like everything else, as we age, maybe their expression will drop over time, and this guy starts building up, and it starts damaging, causing it the damage that lead to, they've linked it to aging, they've linked it to disease, and so forth. Have you ever heard anyone tell you take your antioxidants?
Yes.
That's what the antioxidants will do. It will vacuum clean this guy up. So they're helping the superoxide dismutase get rid of this guy. By the way, that's just one of the bad guys. There are another set of them that are also equally damaging. So they're called radicals, collectively called radicals. So you take antioxidants to help get rid of these things, to protect the cell from damage, long-term damage. So any organism that uses oxygen will produce the superoxide ion, but no worries, because they will also have the SOD, superoxide dismutase, and the catalase. Everybody good? All right. So this slide I'm not going to go over because I actually already did. And let's have some fun with this one. This slide talks about the relationship between 2 organisms. Is it going to be symbiotic or non-symbiotic? This one you'll understand relatively easy. This one takes a little bit of time. Now, in symbiotic relationships, one of the members requires that relationship. In a symbiotic relationship, read it. The organisms live in close relationships with each other, required by at least one of them. Now, in some cases, both of them need that relationship. What I'm saying is the minimum is one of the two organisms must have the other one around. All right, so in some cases, they both need each other, and that would be mutualistic. If you give me something and I give you something and we both benefit off of each other, that's a mutualistic relationship. We both benefit off of each other. So the bacteria that live in your gut, that's a symbiotic relationship between you and the bacteria that live in your gut. So how do they benefit? You're giving them all the food they want. How do you benefit? You get paid back with vitamin K, for example. They stimulate your immune system. They keep your immune system in check, healthy. They help you digest your food. And research has shown that your gut bacteria actually changes your brain physiology. This is big news, actually, huge. Depression, anxiety, and so forth can be associated with an imbalanced normal flora in your gut. So there's a lot of research going on there. And now recent research has shown that it's also involved in obesity. They can predispose you to obesity if it's imbalanced. So there's a lot of push on balancing your gut bacteria for your overall health. And you'll be part of that push with your patients. So that's a mutualistic relationship. It's a give and take. You're offering a home for them, and you're benefiting off of their presence. But then you have this situation, commensalism. Now listen to this one. They can live on your skin. Obviously, they're benefiting from you, but they do nothing to you. They don't pay rent. But at least they're not burning down your home. Do you see that? It's like a guest at your home. He or she not paying rent. They're living free in your home, but at least your home still exists. They don't burn it down when you leave. So that's commensalism. The bacteria that live on you are in this scenario, they're called commensals. Then you got these guys, the ones that actually cause disease. And that relationship is called parasitism. They live in you or on you, so obviously they're benefiting from you, and then they hurt you. So it's like your unwanted guest at home there, living there, the freeloader kind of thing, and then you go to work and you get a phone call, your house is burning. Oh, so that kind of relationship. Do we understand this? All right. So one of the members needs this relationship, must have it. And because of that, that's called symbiotic. Now, in some cases, the other member also benefits from it, mutualistic. Nothing happens to it, commensalism. It gets hurt by it, parasitism. So watch, they both benefit. The host gets hurt, nothing happens to the host. Then you have this one, non-symbiotic. You gotta pay attention to this one. In non-symbiotic relationships, both organisms, they don't need each other. They don't need each other. They don't even know the other one exists. Here's the best example of it. I live in Orlando, and you live in California. I don't know you exist, and you don't know I exist. Why? Because we have, I don't know how far away they are. Let's say 2,000 miles, it might be more. Long distance between here and over there. So I don't know you exist, you don't know I exist. I don't even care that you exist and you don't even care that I exist. Until something happens. What is that? You move in next door to me. Now I have to worry. Worry about what? Watch. Do we get along? Or do we fight it out? I don't know if you do this, maybe it's just me. You see a U-Haul truck pull in. You know the house next door just got sold. Oh, then you're neighbors. And you open the blinds and you say, right? Maybe it's just me. I don't know.
No, it's just not.
Maybe everybody. All right. You open the thing and you say, oh, I wonder, are we gonna get along? Synergism? Or are we gonna go to court, fight it out? Antagonism. So you stay behind the blinds, you get a good look at the neighbors, and you realize, oh, they're nice people. Looks like they have a little kid and so forth. I have a kid the same age. Oh, let me go say hi. And you go say hi, you knock on the door. And they open the door, you say, hi neighbor, how you doing? How do they respond? Get out. Get off my lawn. If you come again, I'll call the police. Uh-oh, antagonism. You see what I mean? Versus, hi, how are you? You bring a cake with you, you eat it together, and you have a party. Heineken involved. Good? And that's synergism. We got it?
Got it.
That's the difference. Did I care before that? No. The only time I cared is when they moved in next door. Right before that, they were all the way over there. I had no idea they existed. Here's another example of synergism. When my neighbor moves in, I'm cutting my lawn on Saturday morning, let's say, with this old beater mower, because that's what I've been using. Before he moved in, that's what I've been using. And it does the job. And then I look over, this guy's pulling out this huge lawn mower with 4 speeds, and oh wow, this thing looks really cool. He cuts his grass in 2 minutes, he's done. Can I borrow your mower? Yes, synergism. Get off my lawn, antagonism. You see what I mean? That's the idea. So the getting along versus the fighting. Now, in the world of bacteria, the fight, if there is a fight, is all gonna be for food. It's all about food. So let's play a game. Watch this. You're gonna be part of it. You can take this, put it over there for me. Good, because we don't want to drop it. Here's the food. Sounds good? No cheating. Be careful. Make sure she doesn't cheat, right? We're gonna fight for this food. You might want to empty your hand there. And we're gonna fight for it. All right. So on the count of 3, I'm gonna say 3, 2, 1. At 1, whoever gets the marker, food, gets to live.
Okay.
Sounds good? That's fighting it out, right? Watch this now.
I'm nervous.
Yeah.
Okay.
You got it.
Ready?
I should say 3, 2, 1 because you're gonna know.
Okay, you say it. You say it.
3, 2, 1.
Oh!
We had that.
She killed me. She killed me, right? That's antagonism. In the world of bacteria, she killed me. That's antagonism. Let's do it again. You think it's gonna happen again?
Yes.
I got something up my sleeve. Watch this one. Go ahead.
3, 2, 1.
And antibiotic, kill. Get the She's like, I'm not gonna let you win. There's no way. So in the world of bacteria, what they do is they release things. I can't afford losing. Because I know what happened the first time. I let her win the first time, by the way. Anyways, listen, I can't afford losing. Because what happened is I died. So I have a trick up my sleeve. I release an antibiotic, kill my competition. Is she here anymore? No, the food is all mine. You see that? The food is all mine. Now I don't have to release an antibiotic. I can release something else, like for example, pH. Let's say she doesn't like a pH of 4. And I have a way to change the pH. That's what I will do. I will drop the pH to 4, destroy my competition, and grab the food to myself. That's called antagonism. This is happening in your gut right now. If you're sitting here today thinking, hopefully, that you are healthy, everything is good, yeah, the bacteria in your gut right now, through antagonism, is keeping you healthy. How? When you eat something that has a pathogen in it, that's what it's going to do. It's going to look at the pathogen and it's going to say, what are you doing here? This is my home. Kill the pathogen. So now you can walk around and say, I feel good, everything is okay. Through antagonism, that's why you feel good. Remember this, it's going to come back up in another unit. So you are healthy sitting here today because of the antagonistic behavior of your normal flora against invading pathogens. So think about it. You have a lot of them, a lot of normal flora in your gut, and a few pathogens come in, they're gonna wipe them out. But you benefit from that by staying healthy. Okay? The biofilm. An example of synergism is biofilm. So remember, synergism is now we're getting along. There's no killing and all that. We're actually getting along. So I don't need her to survive. Remember, before she moved in, I was cutting my lawn just fine. But when she moved in, I got really jealous from her lawnmower, right? So I asked her to borrow it. But was I able to cut my lawn before she moved in? Yes. But now I can cut my lawn better, faster with her mower. You catching the difference here? In synergism, we're going to cooperate with each other for the better. But again, I don't need her, but it's better to have her around. So this one here would be called the pioneer bacteria. This basically means the initial bacteria that infected you. This could happen in your lungs. This could happen in your inner ear. It could happen in the prostate gland of males. Those are the most common locations for the formation of biofilms. Prostate, lungs, inner ear. So this would be referred to as the pioneer bacteria, the initial bacterial infection. Now, if you don't take care of this one at this stage, and it starts progressing, by the time it gets to the biofilm stage, good luck trying to get rid of it. You can't. You won't be able to. And I'll explain to you why. So what happens is this bacteria secretes a glycocalyx. You should remember what that is. You were just tested on it, right? Slime layer and capsule. Those are your glycocalyx, right? So basically an extracellular material. It's secreting extracellular material that is sticky. That's why it's called a slime layer. It's sticky. So any bacteria in the vicinity will get trapped within that sticky material. So if you notice from the picture, initially it's these pink rod ones, right? The pink rod ones are still there, but now you have the caucus ones. Fast forward a little bit more, oh, you got blue ones now. You don't just have one bacteria. You have multiple populations. We call that a community. You now have a community of bacteria, not a single population. Do you live in an HOA community, God help you?
Oh, yes.
Yeah? I do. I think everybody else does, yeah? I can't get away from it here in Florida, right? So you live in an HOA community, you wake up Saturday morning, you have plans to go jogging, swimming, shopping, watch a movie, whatever, and just stay on your couch, do nothing. And you get a letter from the HOA, no, you're cutting your grass. What? Cut your grass or we'll sue you.
Oh, yeah.
Yeah, because you live in a community. And our community needs to look nice. And your grass is not looking nice. But I wasn't planning on it. Well, then change your behavior. Look at that. That letter that you get from your HOA will change your behavior because you now live in a community. Same idea here. The bacteria that live within this community will not behave like they would if they were living by themselves, alone. You know, if you live in an area that does not have HOA, you want to cut your grass, you cut your grass. If you don't want to cut your grass, you don't cut your grass. Nobody's going to say anything. I live in an area where across the street from me is not non-HOA. These guys can do whatever they want. Meanwhile, I'm home cutting my grass because I got to follow the rules. Do you know what the HOA would do to you if you don't follow their rules? Charge? They won't charge, they'll take your home. They can take your home. You signed a piece of paper. And among all the 100 pieces of paper when you bought your home, there was one piece of paper that says, I promise to follow the rules. And if I don't, go ahead, take my home. They can force sell your home to pay for the charges, the fees that were accumulated because of your violations. Watch out. When you get that letter from the HOA, just cut the grass and get it over with. You see? So they send letters for everything. You just have to follow through. Now, it's the same idea with this one. The bacteria that live in this slime layer here, biofilm, they have to cooperate with each other. They have to do things that they normally would not do on their own. That's my analogy here. For the sake of the community. So what are they doing? They're cooperating with each other against you, the host. They want to survive, so they're going to do something together. They're going to gang up with each other against you. That's one reason why they're hard to kill. Because you're not dealing with one enemy, you're dealing with multiple enemies. Number two, the glycocalyx, the slimy layer. Even if you take an antibiotic, the problem, the antibiotic cannot reach the bacteria. This slime layer, it's too thick. The antibiotic cannot go through this very thick extracellular material. That's similar to you trying to swim in a river of honey. Good luck. You're going to have a very difficult time going to the other side of the river. Very difficult time. It's too viscous. So it's the same idea. Now, you got to understand something. The antibiotic can kill the bacteria, but the problem is it can't get to it. That's the problem. So there is research going on today. They're trying to see if there's a way to first get rid of the glycocalyx and then follow through with an antibiotic. Very clever. But they're still working on it. Questions? Yeah.
This is not the bacteria in your gut. This is something that is.
This would be considered an infection.
Yeah.
That's an infection. If you have chronic lung infection, it could be caused by a biofilm that you cannot get rid of. Ear infection, prostate. Now the other biofilm, I forgot to mention, the other biofilm that's very common between all of us here is plaque in our teeth. That's a biofilm too. But in the morning, every morning you wake up, you're removing most of it, but over time it does build up and it hardens and it becomes plaque. That cannot be removed with your toothbrush. It has to be professionally removed through a dental hygienist. Yeah.
What would cause the bacteria to escape from the last
Oh, yeah, it's good. From time to time, one of them can flake off at any given time and set up another biofilm somewhere else. Now you'll have two biofilm infections. Good luck to you. And they'll both be antibiotic resistant. All right. Let's switch gears. Bacteria does multiplies by binary fission. Remember I mentioned this before. There's no mitosis, there's no meiosis, there's no nuclear, thank God. Centrioles, centrosomes, prophase, metaphase, anaphase, none of it is just binary fission. It's very straightforward. Is there DNA replication, however? Yes. So that's the common thing between our cells multiplying and their cells multiplying. Of course you have to multiply, copy the DNA. I mean, at the end you're going to have two cells. What happens if you only have one copy? Then one cell will have DNA, the other one will have no DNA, and what will that do? Nothing. It will die, because it has no instructions at all. So DNA is your instructional molecule there, your information molecule. So the first thing that's gonna happen is the bacteria will copy its DNA. That's no different than our cells multiplying. But then the rest of it is simple. All you have to do is put one copy on one side, another copy on the other side, split the cellular plasm. We're done. Under the right conditions, meaning with nutrition, right temperature, oxygen if it needs it, all the things that we just talked about. Under the right conditions, this can take place in 3 minutes, easy, easy. Within 3 minutes, you have a duplication event, easy, even shorter than that. So they can multiply real fast. It's not a surprise that if you walked in here at 10 o'clock, for example, with 100 cells of bacteria in your lungs, let's say, by 12 o'clock, how many will you have? We'll calculate that. Sounds good? We'll do a calculation for you because you need to know that formula. So you'll be surprised how fast these guys can multiply. But in a laboratory setting, we can follow the growth of bacteria. All you have to do basically is take a test tube with media in it or a flask or whatever, inoculate it, and every once in a while take a sample of it and count how many cells are in the tube. So you're incubating it. And every half an hour or so, take a sample of it and figure out how many cells are in the tube. And the longer you incubate, of course, they start multiplying, and then you start growing them. And when you plot the data, it will look like this. Time of incubation versus the number of cells. All bacterial growth curves look like this. And that's another thing that you cannot leave microbiology without knowing this thing. You can't. So, initially, there'll be no multiplication, no binary fission. So let's say, for example, at 10 o'clock, I take a tube, I inoculate it, I put it in the incubator. No division for a little while. What they're doing is they're acclimating to the new temperature, they're copying the DNA, but the cell itself has not divided yet. And this thing is looking for cell count. So let's say I started with 10 bacterial cells. All along this, I still have 10 cells. But by this time, they all have duplicated their DNA. Sounds good? Then they divide. And that's why you see the upstroke. Once they start dividing, it just takes off from there. This is called the lag phase. Lag means slow. Why are you lagging behind? Why are you slow? So lag phase is because there's no binary fission. All the cell is doing is what? Multiplying its DNA. Then it multiplies and it takes off. Takes off means log phase. Very rapid rate of binary fission. And then a little later, as the bacteria starts consuming the food, they start dying. Some of them. So you're going to get to a point where some are dying, but at the same time, some are multiplying. So 50 cells died and 50 cells were born to some extent. So are you going to get any decrease or increase? No. And that's why it's called the stationary phase, flat phase. No growth and no death either, even. But then eventually, they will really outgrow the food, and even the dead cells in the tube will release things that cause the other cells to also die. So you're going to have more death than living ones, and that's called the death phase. And it makes sense. So during which phase do you have the highest rate of binary fission? Log phase. During which phase do you have the lowest rate of binary fission? Zero rate. Lag phase. During which phase do you have where binary fission and death are equal? Stationary. Death is more. Death phase. Pretty straightforward? Good. This is the formula. So let's try it out. We start out with 100 cells. Let's say you came in with 100 cells in your lungs at 10 o'clock. And let's pretend it's 12 o'clock right now. I know it's 12:23, 4? But let's pretend it's 12 o'clock to make the math easy. The period of time is 2 hours. Do you agree? Yes or no? Yes. The duplication happens every 15 minutes. But you have a lag phase of 15 minutes. How many cells will you have at 12 o'clock? Well, it's 2 to the n initial. Let me do it again. 2 hours is the full length, 2 hours. You start out with 100. Start out with 100, that's this one. The n initial is 100 in my example. What you want to find out is the little n, 2 to the power of n. Well, the little n is the number of divisions. So I have to give you information about it, 2 hours, and every 15 minutes it divides. But it has a 15-minute lag phase. So what would the answer be? Meaning, what would this number be? It is 7. How did we do that? Well, in 2 hours, how many 15 minutes are there in 2 hours? 8. But the first 15 minutes is lag phase. There's no duplication. Watch out for that. There's no duplication. So you don't count that. This little n is for the number of generations. How many times it multiplied? Well, it did not multiply until the 16th minute, right? From 16 to 30 minutes, that's the first multiplication. Now, sometimes I will do that. Sometimes I'll give you the lag phase, time. But sometimes I won't even give you that, so you just assume the whole time is duplication. You see what I mean? Let me give you one. The same exact one. 100 cells, 2 hours, 15 minutes. What would this number be now? There's no lag time. It would be 8. Do you know what 2 to the 8 means? It means 2 times 2. How many times? 8. So let's try this. 2 to the 8 times 100. Do I need a calculator? I hope not. 2 times 2, tell me. Times 2. Times 2. Times 2. Times 2. Times 2. Oh, one more. 128. One more.
256?
256. That's if this is 8, right? 256 times 100.
256. Or 25,000.
25,600. Yeah. So look how that is. I come in at 10 in the morning with 100 bacterial cells. 2 hours later, I have 25,600. Yeah, that's why I'm sick. And I'll probably be coughing and have fever or God knows what else. And I was nice when I said I came in with 100. Can you imagine if I came in with 10,000? What the number would be? You can figure that out. So we'll do some of these, all right? You need to know this formula. It's not that difficult to do. You just need to know what is the initial number. Yeah, you plug it in here. And how many generations, how many times did the bacteria multiply? And you plug it in here and you do your calculation. On the exam, there's no calculator. So on the exam, what I'll be asking you for is the setup. I'll say, which of the following would give you the correct answer? So an example like this, the answer was 2 to the 8 times 100, am I right? That would be one of my choices. Let's say choice B, 2 to the 8th 100. Choice A, 8 to the 2nd 100. You see what I mean? Choice C, 100 to the 2nd times 8, and all the different permutations of it. But there's only one scenario that actually gives you the right answer. So you're not going to have to calculate, but you have to be able to see the right setup that would give you the correct answer.
So the real math is just,
For just practice.
well, like the real math here is just calculating like the exponent n. Like you just have to be able to
Exactly, technically
understand
speaking.
what's going on and then know
Yeah.
that number.
Technically speaking. And the setup.
Right.
What was the two again?
Two, oh, I'm not gonna answer that. You tell me. Why do they use two?
Because multiply, right?
Huh?
You say it's multiply
Well, because bacteria multiplies by
by
binary fission. It's a duplication event every time. If you start out with 10, the next generation will have 20. The next generation will have 40, 80, 160, and so forth. It's a duplication. That's why they use two. Listen to me, please. Let's finish this before you leave. How do we calculate the number of cells? I mean, how do we count the number of cells? Remember in the previous graph, there was listen to me please. In the previous graph, there's a growth curve, right? But you have to be able to count the cells so you can produce the curve. So the data, how do you get the number of cells? You start out with a tube, you inoculate it, every 30 minutes or so you take a sample of it and count the cells. That's what I've been saying. But how do you do it? There's a machine. That's one way. The machine is called the spectrophotometer. Say that 10 times, right? Spectrophotometer. Every lab has one of these. And it's very simple. Watch. Here's a tube that does not have, listen up please. I'll make you teach it. There's a tube that does not have anything in it. That's your blank tube. And there's a tube that has bacteria in it. How do you know? It's turbid. You see? It's the whole idea. You're going to take this tube, put it in the machine to zero it out. We do that all the time when you weigh yourself. When you weigh yourself, you stand on the scale and you keep zeroing it out. Make sure it's zero. I don't want one single extra ounce, right? And then you stand on there to get your weight. Similar idea. You zero out the machine with this guy. Then you put your culture tube in there. And it records something called optical density. OD, optical density. It's basically a fancy word for how cloudy is it? That's it. The cloudier the culture is, the more bacteria it has. That's why it's cloudy. Why is this guy cloudy? Because it's got bacteria in there. So it goes hand in hand. The cloudier something is, the more bacteria it has. And it gives you the number. At 30 minutes, it gives you, let's say, 0.03. Okay. At an hour, it goes to 0.3. Okay. And so forth. And you follow it. And you'll see it actually get cloudier and cloudier and cloudier as the time progresses. You record all the numbers and you graph them. So that's one way. These are numbers that are graphed using the optical density from a spectrophotometer versus time. Is there another way of doing it? Of course. There is this one. Now this one is a little bit more involved here. You can follow the growth of a bacterial culture, but from time to time take a sample of it and plate it out. So at 60 minutes, take a sample of it and put it on the plate. Let it go for another hour, take a sample of it, put it on the plate. Let it go for another hour, take a sample of it, put it on the plate, and so forth. Incubate all the plates. Now you can tell this is a little bit more involved. Incubate all the plates and count the colonies on each one. And they're called colony forming units. And that will give you an indication of how many cells were in the original tube. And thank God we're not calculating any of this stuff. Thank God. And then there's another one. This is for high throughput. This is the last slide. High throughput. If you're working in as a medical technologist in a laboratory analyzing, I don't know, urine samples or whatever it is with bacterial cultures or blood samples to count different types of blood cells and so forth, you're not going to sit there and count each one by yourself. You're going to go put it through a machine. It's called a high throughput machine. It's actually really fancy. It has a carousel. You can take the samples from the patient, put it in the tube, take a sample from another patient, like that, hundreds of them. Press start, go to Starbucks. Go somewhere, do something. come back, all your data done. And you just report the data, that's all. This thing is amazing. Look what is, by the way, you can't just simply use it. You gotta be certified to use this thing. Watch this. The sample, meanwhile, while you're at Starbucks, the sample is going through this very narrow channel. They're very expensive machines. Thinner than your hair. So your cells, they have to all line up one after the other, like this. And that's the whole idea. You want them to line up because there's a laser right here. And every time you break the laser, which would be a cell passing through, it counts. Broke the laser, 1 cell, 2 cells, 3, 4, 5, 6 cells, 7 cells, 8 cells. Very fancy. Do you have an automatic garage door opener at home? You have one of these at the bottom of your garage door. I don't know if you know that. There are 2 metal things. I found out the hard way. My son loved, when he was younger, he loved to kick the ball in the garage, soccer ball. While I was at work, he kicked it, he hit one of those things. He didn't tell anybody. I came home, pressed the garage door open, it opened. I walked in, I closed it, it closed, open. What? Closed it, closed, open. I'm like, what's going on? I thought I was gonna call a garage guy, and I'm not that kind of guy. I have to do it myself. So I got down there, oh, wait a minute, they got misaligned. These 2 things have to be aligned perfectly because one shoots the light and the other one captures it. And the whole idea is this, it's a safety thing. Let's say you close the garage and you realize, oh, I forgot my purse. And you walk back while the garage door is closing, it realizes you just walked through. Why? Because you broke the light. It's a safety thing. If this thing is not working for you, you should fix it because it's dangerous. So it's the same idea. If you interrupt the laser, in the case of the garage door, it opens it. Here, it counts. Very fancy machine. In the laboratory setting, we have this one. This is the last one. In the laboratory setting, you may not have to go to this extreme. I'm only counting 1 sample. We can put it on a very fancy slide with a metal grid that's computer printed. These slides are expensive. And you put your sample on here, and you literally have a counter in your hand looking through the microscope. Don't close your eyes, don't blink, count. You're counting all the cells in this box. Then you count it again, and again, average things out and do your calculation. Thank God we're not doing this calculation either. And this is called a hemocytometer. So these are all the different ways of counting cells to be able to produce the growth curve. Everybody good?
Yes.
Questions?
No.
Tired?
Yes.
Go home.
For this one, right?